Hi, this is Mr. Miller, and today's lesson is on mutations. If you would go to day 5 in your protein synthesis folder and open up, if you would, the mutations note sheet right here, you will find this worksheet right here. And let's define mutations first. So if you would put a text box up there at the top, you can type in a mutation is a permanent change in the DNA. Most important thing to get out of that is we learned that DNA replicates, and when it replicates, sometimes errors occur, and those errors are called mutations. So the important thing to note is that DNA damage or changes are what mutations are, not any changes in the RNA or the protein, but the DNA itself. Okay? And the first thing we're going to look at is something called a point mutation. And this is when only one gene is affected. You can kind of point at the actual change in that gene. Now, we're going to talk about two kinds of point mutations today, really. One is called a substitution mutation, and the other is called a frameshift mutation. So I'm going to zip over to my note slides for a moment, and you can fill in the blanks on this definition of the mutations here. So we've already defined what a mutation is, change in the DNA. And I do want to make sure that you understand how to use the genetic code tables. There are two of them. One of them is on your note sheet, and it's this box one. And of course, you only read the messenger RNA code because you are pretending to be a ribosome translating the messenger RNA into an amino acid. So, for example, if we had UUU was on the messenger RNA, you would look at the first base U, second base is U, so we know we're in this box, and the third base, it says over here, is a U, so you come across that row. UUU is codes for the amino acid phenylalanine. Now, if you prefer the wheel code, I like that better. If you'll notice, it's easy. You start in the middle, work your way out. So if you had UUU as a codon, then you start in the middle. U, work your way out. U. It's still phenylalanine. It gives you the same answer. If you like this code table better, I've put a copy of it in the day 5 folder as well. Okay. Now, we're going to take a look at what substitution mutations do for us here. And so what I'm going to show you is if we had a DNA molecule with this code, we would find that this is the transcribed messenger RNA. So G would bind to C, T to A, C to G, T to A, and A goes to U because we're building a messenger RNA, and there's uracil in RNA. Now we're starting here. It turns out that the ribosome, when it reads the messenger RNA and translates the messenger RNA code into a sequence of amino acids, the RNA has to start at AUG. So it reads AUG, and if you look that up on your code table, you'll find that that codes for the amino acid methionine. UCC is a messenger RNA codon that codes for serine, GAA, glutamic acid, AGC, serine, and CCG, proline. And then UGA is what's called a stop codon. That tells the ribosome to stop making the protein. So in your notes, we define a substitution mutation as one nucleotide is substituted for another. I put in red the words to fill in the blanks. And oftentimes you'll find that the DNA gets repaired by a spell checker enzyme. If it is repaired, there is no mutation. But if it's not repaired, a mutation occurs, and that could lead to a change in the amino acid sequence, which could then affect the protein. That can affect affect a trait. So we'll talk about those types of things that could occur. Okay. Now I'm going to stop this here and go back to your note. Okay, what I would like you to do is pause the video and try this out. Let's suppose this is your DNA molecule, one strand of it across the top row. We know it's DNA, it's got T's in it. Make the messenger RNA that would be created by the enzyme during transcription in the nucleus. So that's a T. For example, if you put a text box in there, the T would bond to an A, then A would bond to a U. Why that went there, I don't know. And then C would bond to a G. So go ahead and do that for each of those boxes and create your messenger RNA. We've broken it into threes to show the codons. Then I want you to use your genetic code table, either the box or the circle, to figure out what amino acids are coded for. So if you look up AUG, you'll see that that codes for methionine, and I will abbreviate with 3 letters. So finish that table, then start the video again. All right, let's check your answers here. Hopefully, when you transcribe DNA into messenger RNA, you found that T went to A to U, C to G. So you should have gotten UCC, GAA, AGC, CCG. And this messenger RNA would leave the nucleus, go to a ribosome, and the amino acids that the ribosome would put down in the correct order would be methionine first, then serine. GAA codes for glutamic acid, AGC coded for serine, and CCG coded for proline. So hopefully you got those correct amino acids put down. Now, that's called protein synthesis. Let's call the DNA up here the normal gene. Now let's see what happens if a change occurs to the gene. So in the above problem, what if the DNA letters AGG here were mutated to TGG? So I'll just put that here. I don't understand why my letters come out of order from what I type, but there they go. Okay, so what would your mRNA be now? Well, TGG is a DNA piece, so ACC, crazy, I don't know why that does that. It's a mutation. Okay, ACC would be what this RNA right here would be. Instead of UCC, it's now ACC. So what amino acid does that code for? Go ahead and look that one up. Hopefully you looked up ACC and found that ACC codes for the amino acid threonine. So we'll just type that one in. You can just write the first 3 letters if you want to. Threonine. Now, let's take a look. I'll go back to my slideshow at what kind of mutations can occur. And suppose we had a mutation and that T that I'm pointing to here was substituted. If you'll look at the normal gene up here, you had an A and it gets replaced by a T. Well, that affects then the messenger RNA that we're building, and it affects the protein that you built just now too. So the protein you just built would have had all of the amino acids correct except for threonine replaced the serine. So what we find is a substitution mutation where one nucleotide, A in this case, was substituted for a T, ended up producing one amino acid different in this protein. Okay? Now sometimes a base substitution has no effect on the protein at all. And if that's the case, we call it a silent mutation. So you can write that in your notes. Sometimes a silent mutation doesn't even change the amino acid. For example, here we have GTC, TAC, that's all the same in this changed one, but here we've got AGA instead of G, so an adenine replaced this guanine, if you can see that right there. And as a result, it did change the codon, but the codon coded for the same amino acid, serine. So as a result, it's the same exact protein. But sometimes a single base substitution can be very serious. So hemoglobin, which is a protein in your blood cells, is made from 574 different amino acids make up that protein. Sickle cell disease is caused by one amino acid being incorrect. So in this case, if you look at the normal cells have normal DNA, they produce normal RNA, and the protein sequence that we get is as follows. So if we had this highlighted adenine put in instead of a thymine, that seems like such a minor mutation. This thymine is substituted with an adenine. But with that change, notice that what we get is a change from GAA to GUA for one of the codons. The only amino acid affected is that codon's amino acids, which turns a glutamic acid into a valine. That one change is in a critical spot in the protein, and it makes hemoglobin malformed, which multiply that by millions of hemoglobins, and they stick together, and they cause the cells to form a sickle shape, like the farmers used to use back in the day, sickles to cut the hay with. And that's where the name of the disease, sickle cell anemia, comes from. So to review that, one changed base, T here, CTT, is changed to CAT. That changed one amino acid. That causes sickle cell anemia. Now, substitution mutations that we just talked about can be very minor. They can be silent, or they can cause a disease. Really bad mutations are called frameshift mutations. So if we take that very same problem, same gene, and we insert or delete a base, then the entire sequence of the DNA, and thus the RNA, after that mutation gets shifted. We'll talk about that. It's much more serious to the structure and function of the final protein. And stop codons can be lost. A protein is usually non-functional because of all the incorrect amino acids put in. So let's look at a couple, then we'll look at our note sheet. One of the things that can happen for a frame shift is a deletion. So let's imagine, I've just copied the gene from above here. This thymine here, let's just say it gets deleted. And if it did, the cell will produce a messenger RNA that used to have 3 A's in a row, but now it's only got 2 because we deleted that. So now the next base is C goes to G, G goes to C, G goes to C, and so forth. As a result, any amino acid past the one where the mutation is going to be wrong because you changed the reading code of 3 bases by 1 letter. So everything is wrong. Even the stop codon is gone here. Now, one other kind of frame shift mutation is when a base gets inserted incorrectly. So once again, this was our normal gene producing a normal protein. And what I did was inserted a base right in here. It's going to go on here. Right here, we're going to see a base get inserted. Insert that thymine right in here. Like say that was a screw up of DNA replication. The mutation is we inserted a thymine. That means when you make messenger RNA, you're going to make another adenine here to fit that in. So that cytosine binds to guanine, guanine, cytosine. But again, it screws up the 3 base sequence. So now, when you make your proteins, the first 3 are fine, But now you've added that base in there, so we end up with a different set. We lost our stop codon, and we continue down the line with all kinds of different amino acids. So the protein being produced is very much changed and most likely non-functional. The one last mutation I want to mention is a chromosome mutation. And they're called translocation. That's not important yet. What is important is to understand that chromosomes have lots of genes, and sometimes chromosomes with one, like this one, number 5, with one set of genes on it, they can end up being moved or translocated to a totally different chromosome. So here's the terms to fill in on your note sheet. But the point is, we had a segment of chromosome 5 break off and move and attach to chromosome 8. The bases may not have been put in the correct order, but many genes could have been cut off here and put here, and that affects how they're read. So from this, all I really want you to know is that sometimes a mutation can happen with lots of genes. Instead of just a point mutation that affects one gene, this may affect 100 genes. Now let's take a look at the impact on offspring. One type of mutation can happen in a skin cell or a nerve cell or a blood cell. Those are called somatic mutations because they happen in our somatic cells. The good thing is you can't pass those on to future generations. So this gentleman looks like he's been out in the sun too much, got a nasty sunburn. And so what he found out was he got some cells damaged. They could actually have mutations that cause the cells to reproduce out of control, and he could get skin cancer. We all know being in the sun too much can give you skin cancer. But of course, that didn't affect his gametes, his sperm cell, and therefore he can't pass that on to his kids. Those mutations that happen in your skin cells or other somatic cells don't get passed on to your kids. If you have a mutation in a germ cell, which are your sperm or your egg producing cells, don't worry about these terms, we'll come to these next unit. But diploid cells that undergo meiosis, make our sperm and egg, they can be passed on to future generations. So what I really want you to know from this is mutations that happen in a sperm or an egg are going to be passed on to your child, and then every cell in their body comes from that fertilized egg. Therefore, every cell in their body will have that mutated gene in it. So we often worry more about mutations that occur in the sperm or egg. Now, causes of mutations. We know that sun or cigarette smoke can cause mutations, and these things, whether they're chemicals or energy from the sun, are called mutagens. And a mutagen is anything in the environment that can change your DNA. It can either speed up your replication process, it can break apart nucleotides. For example, ultraviolet light from sunlight can break these hydrogen bonds between the A and the T and obviously cause damage. You could replace an AT base pair with a GC base pair. So one thing you can do is practice these questions here. And the next thing I'd like you to do is to work on the practice mutation sheet. So for this practice sheet, what I would like you to do is pretend this is the original normal gene here. You're going to transcribe it into messenger RNA, so T would go to A to U, and so forth. Then use your code table to find out what amino acids are coded for. Once you've done that, they're going to say, hey, let's see if a mutation happens here, what the effect will be. And so you'll notice this one looks like it has a T here instead of a C. So you compare it, always go back and compare it to this GNA. Come up with what mRNA will look like, what would the amino acids look like, and then circle one of these. Was it a substitution mutation, an insertion, or a deletion? And then how did it affect it? Was there no change in the amino acids, one amino acid changed, and so forth? So circle one of these. And they're going to ask you to do that for a number of different mutations. So work that practice problem, and there is an answer key up on Schoology. So work that, and I hope this helps.